In the current issue of New England Journal of Medicine on May 9, 2024, there were 4 original articles, and out of the 4, 3 are from the hematology. Now here the 2 studies are for the cure of sickle cell disease and beta thalassemia respectively, and the third one is the new treatment modality for immune thrombotic thrombocytopenic purpura. So I would try to explain the basic principle of these studies. So for sickle cell disease, FDA has approved 2 autologous cell-based therapies. So what is autologous cell-based? We will talk about it later, but first try to understand these 2 therapies. Of the second one, I will talk about second one right now. Hexagamglogin autumn T cell Cas-GV is a gene editing. This is gene editing technique. This is the same technique. This is the same treatment which are we going to talk about in the New England Journal of Medicine, the 2 studies that published in this issue of May 9, 2024. Again, this is the gene editing. We are going to edit the gene. Now, another therapeutic modality is the lovotibeglogin autumn T cell. This is the gene therapy. So gene therapy is different from gene editing. Gene therapy is different from gene editing. So lovotibeglogin autumn T cell, this is gene therapy. And here hexagamglogin autumn T cell, this is the gene editing. Now in gene therapy, because this is not the main topic for today, gene therapy, what we do here in gene therapy, the basic aim we are targeting is this, that we have to express hemoglobin A, T87Q, hemoglobin T87Q. This hemoglobin A is resistant to polymerization. So how we will do it? We will do with the lentiviral vector BB305. We take the hematopoietic stem cell of the patient and we treat it in the laboratory. We do the genetic manipulation through lentiviral vector BB305, and that express HbA, hemoglobin A, T87Q. So this hemoglobin is resistant to polymerization. This is the basic principle of gene therapy. Now, what is the principle of the gene editing? This is the same principle that is going to be used in our New England Journal of Medicine study. So here, what we do is this, that we disrupt the transcription factor BCL11A. So this thing is extremely important here to understand further. BCL11A is a transcription factor that converts hemoglobin F to hemoglobin A. So here in gene editing, we disrupt this BCL11A, so then hemoglobin F will not be converted into the hemoglobin A, and so we will have more hemoglobin F. So here in gene editing, we are aiming to get more hemoglobin F. Our goal is to get more hemoglobin F in both sickle cell and thalassemia. Now let's try to understand what is autologous stem cell transplant. Now in this study, what we do, we will often hear about autologous. So what is that? So what is autologous stem cell transplant? So how they do is this, that the patient that we want to treat with the gene editing or gene therapy, so we give him GCSF for 4 days in a row. The basic principle is 4 days in a row. This dose is of the multiple myeloma, from my notes of multiple myeloma. But the same principle applies here. So we give GCSF 4 days in a row, and that would produce the stem cell to make more CD34 positive cells. Now we give GCSF for 4 days, and then on the 4th day we give patient this medication, Mozobil, Plerixafor. So what this medication does, this medication basically pushes the stem cell to come more into the blood so that we can collect more stem cells. Then we check if there are enough CD34 positive cells in the blood. Then we do apheresis and we remove CD34 positive cells. Now here we have collected our stem cell from the patient, and now we have our stem cell. So now we will treat this stem cell in the laboratory. Okay, so we can treat it with either gene therapy or gene editing. So again, understand the first try to understand the thing. The thing will whole start with the patient. First, we try to get the stem cell. We'll give the GCSF. We will on the fourth day we will give the mucobil, and then we will collect the stem cell. And now we have the stem cell here, and now we send the stem cell in the laboratory for the manipulation with the gene editing, and then meanwhile we will send the patient home because we have to prepare the stem cell. Now let's talk about how we prepare the stem cell for our particular study for the gene editing. Now the principle that is used in the gene editing is called the CRISPR-Cas9 complex. So what is CRISPR? Clustered, regularly interspaced short palindromic repeats. And this is Cas9 cluster CRISPR associated complex 9. So the whole idea of CRISPR-Cas9 came from the bacteria, like how bacteria develop their resistance when they are attacked by the viruses. So what the bacteria do when the viruses attack the bacteria, those particular viruses that attack the bacteria, they are called the bacteriophage. So when the viruses attack the bacteria, so what bacteria do? They develop resistance. So how they develop resistance? They break the nucleus of the virus that is attacking them. They keep the nucleus stored in their genome, and when in the future the same virus attacks the bacteria, that DNA will make the RNA in the bacteria. Now that RNA will be loaded with a protein that is called the endonuclease. Now this RNA that was made by the genome that was already stored, and now this RNA is loaded with the protein. Now this protein is gonna be the endonuclease, and now this RNA will identify the enemy virus, and the protein will break the DNA. This is the principle of the CRISPR. That the idea was taken from this thing. Now just try to understand, to make it easy. Just imagine that if a country gets a spy from another country, from an enemy country, they get a spy. Okay, the purpose of the spy was to do the bad job in the country. So what they do, they catch the spy. Right? Bacteria catch the DNA of the virus that was attacking them. So they catch the spy, they get the information from the spy. The bacteria use the DNA of that attacking virus, and now the information that we got from that particular spy whose job was to harm us, we are going to use the same information in the future to attack the virus that if that virus would attack us in the future. All right, so this is just think like that. Now let's apply that principle over here. Now let's come to this diagram. This is the I googled it, and this is some website. Where did it go? Okay, now here this is the RNA this is the RNA. This RNA is called the guide RNA. This is the same type of RNA that was made by the bacteria, and that is gonna attack to the virus. Okay, so this is the RNA that is gonna be that is gonna be attached here on the DNA of the virus of our target, and this RNA is loaded with a protein, Cas9 enzyme. This is an endonuclease protein, and here there is often a DNA that is called the motif DNA that this guide RNA will recognize. So this RNA will recognize the motif DNA here. So then this Cas9 enzyme will just do some breakage here. Now the DNA, when it will try to repair it, it will most likely lose the function. This is the basic principle of CRISPR-Cas9 gene editing process. Again, this diagram, I used it from the website. No copyright violation intended. The purpose to share it just only for educational purposes. If you want to learn more, this is a very good resource. You should go there, and if you put it in the Google, the first thing it will come this. Okay. Now in our gene editing, what we want to do, we want to disrupt the transcription factor BCL11A in autologous hematopoietic stem cell. So now we already collected the autologous hematopoietic stem cell when we gave the patient GCSF for 4 days, and then on the fourth day we gave Mozobil. We collected the stem cell, CD34 positive cell. Now we took that stem cell in the laboratory, and with the help of CRISPR-Cas9 we are gonna disrupt the transcription factor of BCL11A. Okay, so now this BCL11A has been disrupted. So then what will happen? The hemoglobin F will not be converted into hemoglobin A, so we will have more hemoglobin F. Okay, now we are in the laboratory, we are ex vivo, and now we have treated our stem cell. Now what's the next step? The next step is this, that we have to put this stem cell that we have treated, we have to give it back to the patient. Then this will be the autologous stem cell transplant. Okay, we took it from the patient. When we give GCSF, we give Mozobil on the fourth day. We treated it in the laboratory. We give CRISPR-Cas9. We turned off the BCL11A. We have now more hemoglobin F producing stem cell. Okay, now we are in the laboratory, and now the next step we have to do, we have to give this transplant, stem cell back to the patient. So how we give it, let's talk about that. Now we will call our patient and we will tell the patient to come to our suite for chemotherapy, or our hematology suite, because now we have to do the preparative chemotherapy. So preparative chemotherapy, this is called the myeloablation. So what we have to do, we have to kill the bone marrow that is present in the patient. We have to kill it, we have to suppress it, so that we will give our new hematopoietic stem cell that we have treated with CRISPR-Cas9. We want to give it to the patient, right? But before we give the new brand new CD34 positive cells to the patient, we have to myeloablate. We have to, in simple word, we have to kill the previous sitting bone marrow CD34 cell. So how we do? In this CRISPR-Cas9, they use busulfan. Now I wrote here melphalan because this is from my notes of the multiple myeloma. In multiple myeloma, they use this medication melphalan 200. It's a very common mel 200. They call it mel 200. I was looking it up why they use busulfan here in gene editing in this, but I could not find the answer. But definitely there will be some reason. So busulfan, they use busulfan. Okay, melphalan is for the multiple myeloma. Principle is the same, but they use the busulfan. Now when we have give busulfan, after a few hours, then we inject our new treated CRISPR-Cas9 CD34 cells, in which we have shut off the BCL11A gene. We are gonna give this new treated CD34 cell to the patient. We will just infuse it. Now once we transfuse our treated CD34, our treated stem cell to the patient, they go back into the bone marrow. Now the process of going the treated CD34 positive stem cell to the bone marrow, this process is called homing. Now here our autologous hematopoietic stem cell transplant is completed. We have new CD34 cells in which BCL11A has been turned off. We have more hemoglobin F. So cure for the thalassemia and the sickle cell. So this is the short summary. I hope this will help you understand when you will read about these two studies. Very fascinating study. I was so much happy. I was so much passionate to study about it, and it's so beautiful to see that we are able to cure these patients. Excellent. Now let's talk about the third study about the recombinant ADAMTS13. So before we talk about recombinant ADAMTS13, so let's try to understand what is TTP, what is the issue going on. Now this is the cartoon diagram. This is made by me. So this is one Willebrand factor. This is our platelet. Now here we have the endothelial injury. We have one Willebrand factor exposed. Now platelet, here it grows its leg. This leg is actually GP1B receptor, and this GP1B receptor, it comes to the one Willebrand factor and it asks, oh, hey, you need help. And one professor says, yes, I need help. So he puts his legs, he sits with the help of it, like the leg, he establishes his bonding with one Willebrand factor, and then platelet grows its arms to help. These arms are basically GP2B3A receptors. So this is the leg that has been fixed on the von Willebrand factor. Now this is the arms that are hugging the other platelets. Hey, try to help this von Willebrand factor. This guy, he's in trouble. So this is the basic principle. These hands are GP2B3A, this leg is GP1B, okay? Just like the same thing, let's say this is a person who is a good person who wants to help the people who are in trouble. So first he will go to them. He will first go to them. It's not easy to help someone when you are miles away from the person. You will go to him, you will stand with him, you will tell him that, hey, I'm standing with you. You will go to him, and then you will use your connection. You will use your arms, GP2B3A, to call your friends to help this guy. Okay, so this is the principle. Now, what is going on in with TTP? In TTP, the problem is this, that von Willebrand factor is too much clingy. You know, sometimes you try to help some people and these people are like, they are too much help demanding. They don't do anything themselves, right? So von Willebrand factor is controlled by a thing called ADAMTS metalloproteinase 13. Okay, it's controlled by that. And now there are autoantibodies against ADAMTS metalloproteinase. So the von Willebrand factor will not be shut down. The von Willebrand factor will be super active. Von Willebrand factor will always asking for platelet. Come for help, come for help. So the platelets are like sticking, because platelets' job is to help. Platelets are, they want to help this. But the problem is in the von Willebrand factor, and because von Willebrand factor is too much clingy, because ADAMTS, actually the problem is in ADAMTS, not von Willebrand factor. ADAMTS is the problem here. How we treat it, now we do the exchange to remove the autoantibodies, and the plasma, we give plasma to give ADAMTS 13. But now here in this study, they use the recombinant ADAMTS 13.